So right now we know that glucose levels are elevated in the blood. Blood sugar is elevated. And what we need to do is get that glucose or that sugar into our muscle cells. Pretend that this is a muscle cell right here. Okay. How do we do that? Well, we also know that the rise in blood sugar triggered pancreatic beta cells to release insulin. And we know that that insulin that was released by the pancreatic beta cells is traveling through the blood. Okay. And what that insulin is going to do is going to leave the blood, it's going to go into the interstitial fluid around our muscle cells, and then it's going to bind to receptors located on our muscle cells to initiate the process of allowing that glucose that's also within the bloodstream to be able to come into the muscle cell as well. Okay, so here's what we're going to do. We're going to first start off by looking at the receptor that insulin that is traveling within the bloodstream binds to on the skeletal muscle cell. Imagine this being the plasma membrane of the skeletal muscle cell. This is an insulin receptor located on the skeletal muscle cell. Okay, here's the skeletal muscle cell plasma membrane, and this is the insulin receptor, which is a transmembrane, ultimately integral protein. Okay. Oftentimes it's referred to as kind of tyrosine kinase receptors, but it's a little bit more sophisticated than that. So let's talk about what this insulin receptor, this integral protein that serves as a receptor, looks like. What's its structure? So what it has, first of all, are two alpha subunits and two beta subunits. Okay. The alpha subunits are extracellular, meaning that they lie outside of the cell in the interstitial fluid. And the beta subunits, they exist both intracellularly. Here would be the intracellular side, meaning that they exist to a degree in the cytosol. They exist in the plasma membrane, and they also exist to a degree in the extracellular or interstitial fluid. So the four major proteins of an insulin receptor are the two alpha subunits and the two beta subunits. Now, the two alpha subunits are bound to one another through these disulfide bonds, and as are the alpha and beta subunits. Okay. Now, also, you'll see, just for clarity here, there's this glycosylation site that exists on the alpha subunits as well, and that's necessary and needed in order for insulin to actually be able to bind to the alpha subunits. And that's exactly what insulin does. Insulin comes along, in the bloodstream, and what insulin will do is it will leave the bloodstream into the interstitial fluid, and then what insulin will do is it'll bind to the binding sites that exist within the alpha subunits. Okay? And when insulin binds to the alpha subunits, what that's going to result in is the activation of these tyrosine kinase domains that exist in the beta subunit. So let's talk about what those tyrosine kinase domains are located. Let's talk about what those tyrosine kinase domains do. And as I've said before, oftentimes the insulin receptor will be called tyrosine kinase receptors, but in reality, the tyrosine kinase domain is just on the intracellular side of the beta subunits. So there's just certainly more to the overall structure of the insulin receptor than just the tyrosine kinase domains located on the beta subunits, but that's going to be the most important part of the receptor for reasons that we'll discuss right now. So again, insulin binding to the alpha subunits ultimately activates the tyrosine kinase domains located on the beta subunits, again on the intracellular side. And what these tyrosine kinase domains are, well, first of all, look at the name, kinases. Tyrosine kinase domain. So this is actually an enzyme, and the tyrosine kinase domain, what it does is it starts phosphorylating tyrosine residues. Okay. Proteins that have tyrosine residues, these tyrosine kinase domains are able to phosphorylate. And what are the major proteins located inside of the skeletal muscle cells? And this is true of other cells too, but we're focusing on skeletal muscle cells. What are the major proteins that exist inside of the skeletal muscle cells that have these tyrosine residues that these tyrosine kinase domains are able then to phosphorylate? What we have are these docking proteins that are referred to as insulin receptor substrates. Okay? And there's a variety of them. You'll hear them just referred to as IRS. Okay? Insulin receptor substrates. And there's IRS 1, 2, 3, 4. We're interested in IRS 1. Insulin receptor substrate 1. Look at it. Look at its name, first of all. Insulin receptor substrate. So it's a substrate for the insulin receptor, meaning that the insulin receptor is going to phosphorylate it. Okay. The IRS1, insulin receptor substrate 1, has these tyrosine residues located on it. So it can get phosphorylated by the tyrosine kinase domain located on the insulin receptor, and that's exactly what happens. Okay. Let's look at that more closely. Here's a simplified but helpful image. You can see insulin binding to the alpha subunits of the insulin receptor. Okay, and then you can see the two beta subunits here. And when insulin binds to the alpha subunits, it activates, you don't actually see it in this image, but it activates the tyrosine kinase domains located on the beta subunits. And the tyrosine kinase domains, what they do is they start phosphorylating proteins with tyrosine residues. And here is IRS1, insulin receptor substrate 1. Insulin receptor substrate 1 is an intracellular protein that has tyrosine residues, so it can be phosphorylated by the tyrosine kinase domains located on the insulin receptor, and that's what's going to start happening once this insulin receptor is activated by insulin. IRS1, insulin receptor substrate 1, is going to start getting phosphorylated by the insulin receptor, specifically the tyrosine kinase domains located on the beta subunit. Okay, so what you're seeing actually in this image is that IRS1, you can see by the P's, is being phosphorylated. So insulin receptor 1, IRS1, is being phosphorylated, and thus IRS1 has been activated. That is the first step in the insulin receptor pathway, is to phosphorylate and activate IRS1. Now, IRS1 is a docking protein. IRS1 is not something that goes around and phosphorylates things. IRS1 has been activated by being phosphorylated. So IRS1 has been phosphorylated, and thus it is activated. And then what it goes is, what it does is it docks to this other protein, which is PI3K. What IRS1 does is it docks to PI3K, and when PI3K has been docked to, PI3K has been activated. Now, I don't want to go through every single one of these acronyms because, again, it's like you could spend half the time just going through the acronyms. So I'll show you the big part of PI3K. Big thing about PI3K is the fact that it ends with a K, and that stands for kinase. So it phosphorylates things. IRS1 docks to PI3K and activates it. And PI3K, which is a kinase, is going to go around and it's going to start phosphorylating other proteins. So what PI3K does is it goes over to what's called usually PIP2. Some of you have seen this protein before. And what it does to PIP2 is it phosphorylates it. And when it phosphorylates it, a phosphate has been added to PIP2. And what PIP2 becomes is PIP3. Okay, so PI3K phosphorylates PIP2 and turns it into PIP3. And PIP3 essentially is now this active protein that's going to start doing some more. It's actually going to recruit this other protein that's going to be very important to the pathway. So let's talk about that now. All right, so we're at the level of PI3, you can actually see it right here. You can see that it has, if you look closely, you can actually see when PI3K comes over and phosphorylates PIP2, it adds a third phosphate to it, and that's why it becomes PIP3. Okay, so what does PIP3 do? PIP3 recruits and activates this other protein, which is referred to as PDK1. Okay, what is PDK1? The big thing is you see another K here, and that stands for kinase. Okay, so the activation of PIP3 recruits and activates PDK1. And PDK1 is a kinase, so it is going to go over and phosphorylate something. When PDK1 is activated, it's a kinase, and it's going to phosphorylate one of the most important proteins in this entire pathway. Oftentimes you'll hear to the insulin pathway as kind of the AKT pathway. And what PDK1 does is it phosphorylates AKT. You can see it actually right here. Phosphorylate. Let me erase this a little bit. You can see what's happening here is PDK1 is phosphorylating, again, kinase, it is phosphorylating AKT. And when AKT is phosphorylated, AKT has been activated. So AKT actually is what's called protein kinase B, PKB. You can actually see it listed as both things. Okay, so what PDK1 does is it phosphorylates and it activates AKT, which is also known as protein kinase B. Why is that important? Well, because what is a kinase? A kinase is something that goes along and phosphorylates things. So this is the big downstream effect of the insulin pathway, is the actual activation of AKT, or protein kinase B, by PDK1. PDK1 phosphorylates AKT and activates AKT, which is also known as protein kinase B. And now what protein kinase B, or AKT, can do is go around and phosphorylate something else, which brings us to a whole different conversation. Not a different conversation, but something that really we have to deviate from we have to deviate from this pathway for a little bit to make sense of what protein kinase B, or AKT, does. All right, so the way that insulin actually comes in to the skeletal muscle cells is through facilitated diffusion. There are what are called these GLUT4 proteins, glucose transporter 4, or GLUT4. Okay? And you need these GLUT4 proteins they're actually stored within intracellular vesicles. And what you need in order for the glucose that's, traveling through the bloodstream and ending up in the interstitial fluid, in order to get that glucose into the skeletal muscle cells, what you need are these GLUT4 proteins to what's called translocate. You need them to go from the intracellular vesicles and embed themselves into the plasma membrane of the skeletal muscle cell, so that then they can bring the glucose molecules in through facilitated diffusion. The GLUT4 proteins are what function through facilitated diffusion, right? They're initially open, facing the extracellular fluid, interstitial fluid, and closed facing the intracellular fluid, and then glucose comes in and docks, and then it changes its orientation to actually allow the glucose molecule in. But the thing is, with skeletal muscle cells, these GLUT4 proteins are not naturally, they don't naturally just exist in the plasma membrane. You actually have to get them to leave these intracellular vesicles, right? They actually have to leave these intracellular vesicles that they're stored within, and this is called translocation of GLUT4. You need these GLUT4 proteins to translocate to the plasma membrane, embed themselves into the plasma membrane, and then bring glucose in through facilitated diffusion. And the function of the insulin pathway is to trigger what's called translocation of GLUT4. The insulin pathway is what's going to trigger these GLUT4 proteins to actually leave the vesicles embed themselves into the plasma membrane and then bring glucose in through facilitated diffusion. So now the question becomes, what about, if you follow this downstream, what about AKT, or protein kinase B, as it's also called, allows for GLUT4 translocation? What is this protein going to do that enables this to occur? So the question is, what allows for these GLUT4 proteins to translocate to the plasma membrane and bring glucose in through facilitated diffusion? And what it looks like is you need these Rab proteins called Rab gaps to be active in order for this to occur. What you need is this Rab protein to have GTP bound to it. Okay? And that's when we say this Rab is active. Okay? It's when Rab has GTP bound to it, and that's what triggers GLUT4 translocation. Okay? But here's the thing. In a normal state, when insulin is not bound to the skeletal muscle cell, what we have is this AS160 protein that's very active. And when AS160 is active, what it's doing is it's keeping the Rab protein with a GDP bound to it rather than a GTP bound to it. And when Rab has GDP bound to it, it's inactive. Okay? So thus, we would not be getting any GLUT4 translocation as long as Rab is inactive. Okay, so this is, I'm kind of doing this in a backwards way because I think it's actually the easiest way to understand this. In a normal circumstance, when insulin is not bound to the skeletal muscle cell, AS160 is active. And what AS160 is doing is essentially keeping Rab inactive by having GDP bound to it. And as long as Rab is inactive and not active, right, there's going to be no GLUT4 translocation, right? This is not going to happen. Perfect. However, I just told you that insulin has bound to the receptor, and if we follow the pathway downstream, we have activation of AKT, or protein kinase B. Okay, so something is going to change here. AKT, or protein kinase B, we've always known as the most important protein of the insulin pathway for a long time. Exactly what it does, this is the theory that I'm going to show you, but I would not say that this is 100% certain or 100% established by any means, but this is the working theory at the moment. What AKT does, or protein kinase B does, remember it's a kinase that phosphorylates things. What many scientists believe that it does is that it phosphorylates AS160. And when AS160 is phosphorylated, AS160 is knocked out. It doesn't work anymore. Okay, so look at this. It's AS160 has been covalently modified, as we would say, right? You've seen many levels of covalent modification throughout this process. But what you're seeing here is AKT going over and phosphorylating AS160. You see all the phosphates that have been added to it. And when AS160 has been phosphorylated, it is now inactive. Right? It can't function anymore. And remember what AS160's job is. AS160's job is to keep Rab inactive by having GDP bound to it. Well, now that AS160 has been knocked out by protein kinase B, also known as AKT, what's going to happen to Rab is going to become active. If we knock AS160 out, then Rab can swap out GDP for GTP and become active. And when Rab is now active, because it has GTP bound to it, it's going to trigger GLUT4 translocation. GLUT4, this, GLUT4, whatever you want to call it, is going to leave the vesicles, and GLUT4 is then going to embed itself into the plasma membrane, as you can see here, and then it's going to take the glucose within the interstitial fluid into the cell through facilitated diffusion. And now what's going to happen to glucose levels within the blood? They're going to fall. Okay? Blood sugar is going to drop thanks to the activation of the insulin pathway within skeletal muscle cells. And this pathway exists within other muscle cells too, like your adipocytes, your fat cells have this exact same pathway as well, and so do other cells in your body. Okay, this is the primary insulin receptor pathway that enables cells to uptake glucose. Okay. As long as insulin is bound to the insulin receptors on the skeletal muscle cells, okay, you know that AKT, or protein kinase B, is activated, AS160 is thus phosphorylated and inactive, Rab is thus GTP bound and active, and GLUT4 translocation is occurring to allow for glucose to be uptaken from the interstitial fluid and thus the blood through facilitated diffusion into the skeletal muscle cells. Just in case this is helpful, here you can see AS160, as long as AS160 is active, not phosphorylated, it's keeping Rab bound to GDP, and thus Rab is inactive. And here you can see once AS160 is knocked out, right, AS160 is inactive, Rab becomes GTP bound and thus active and drives GLUT4 translocation. This is really just here in case this is helpful for you to see this particular part of the pathway isolated. Now, insulin is degraded very, very quickly. It's degraded incredibly quickly by liver cells. It's degraded also by the kidney. It's degraded actually by it's believed to be degraded by the actual insulin receptors themselves. So insulin only stays active inside of the body for a couple minutes in reality. Okay, so if you're wondering, like, what triggers this pathway ultimately to come to an end, the answer is, well, insulin does not stay elevated in the bloodstream for a very long period of time. And as long as insulin then is not bound to the insulin receptor, there are these intracellular proteins that will assist in removing all these phosphates that have been added to these different proteins, and thus reversing the reaction. If you're curious where this conversation is headed, we're going to talk a little bit about type 2 diabetes. In a basic sense, a disease that we understand very well, but from a detailed perspective, like the intracellular mechanisms that are involved in type 2 diabetes, something we don't understand still to this day all that well. We're going to talk about type 2 diabetes, and then we're going to talk about how exercising, getting these skeletal muscle cells to contract, is very beneficial for those that have type 2 diabetes. And it's going to be through the activation in a different way due to skeletal muscle contraction of this GLUT4 translocation pathway.